Good evening, everyone. Today we are here at the problem solving session number 6 for the course Biointerface Engineering. So, I'm waiting for several other students to join. Till then, we can just go through the first few slides. Okay, so this particular course has the instructor called Dr. Lalit Mohan Pandey. He is the Associate Professor in the Department of Biosciences and Bioengineering, IIT Guwahati. So I myself am Shilpa Nandi. I'm a Prime Minister Research Fellow at the Center for Environment, IIT Guwahati itself. So we will be covering the week 6 assignment today, and we will try to solve particular questions related to the week 6 assignment. And also, this is a doubts and interaction session that is held every week. That it is basically a live session. So this occurs every Tuesday, that is from 7 to 9 p.m. So if you have any queries during the session, any questions regarding the course content or any other questions as well, you can raise your hand or unmute yourself as well as then raise the query. So the questions can also be posted in the chat box, and also there is a discussion forum for the course which is present in the portal itself, uh, naming ask a question, and so any questions related to this live session or the course can also be raised there. So questions will then either be addressed in the forum or in the next week's live session. So for this particular problem solving session number 6, the outline of this live session would be: firstly, we will go through the week 6 course content recap, that is particularly the lecture number 16, host responses to biomaterials, lecture number 17, which is cell adhesion, and lecture number 18, that is biocompatibility of biomaterials. So after we will go through the recap of the week 6 course content, we will then try to solve certain questions and discuss the answer as well. So those particular questions will help you to solve the questions of the assignment number 6. Right. So let's move ahead with the week 6 course content recap. So we'll go through a quick revision of week 6 contents, as we are lagging behind 2 weeks. Already the assignment like course content for week 8 has been also uploaded, as well as the assignment also. Right. So starting with lecture number 16, which is host responses to biomaterials. So in this particular lecture, we first went through what is this cell surface interactions. So the interactions may include surface water interaction, then it will go on to protein surface interaction, and lastly cell surface interaction. Right. So during these cell surface interaction, first there will be adhesion, then proliferation, and then differentiation. Also, during any biomaterial implantation uh, within the body, there can be either two ways by which this biomaterial implantation occurs and leads to two effects: either it causes the tissue damage and or it induces the Roman effect. So in case of tissue damage, it will activate the immune system and will then lead to innate immune response, that is the will be activated due to the iatrogenic injury. Also, there can be temporary matrix formation. So this will again lead to innate immune response due to the presence of the biomaterial in the body. Right. So this inflammation serves to contain neutralize, dilute, or wall off the injurious agent or process. This particular process occurs through a mechanism that is known as signal cascades. Right. So lastly, there will be two possible outcomes. So if the biomaterial is non-degradable in nature, so there will be either tissue integration, there will be foreign body reaction, or even encapsulation. And if the biomaterial is degradable in nature, there can be remodeling, foreign body reaction, and fibrous scar. So the host cell response, likewise, I said, can lead to tissue injury during implantation, or there is a particular biomaterial implantation. So tissue injury during implantation would result in the wound healing processes, which is the host cell response. And in case of biomaterial implantation, there will be persistent inflammatory responses. So this leads to hemostasis. So firstly, what happens? There is a vascular spasm, or there is the constriction of a blood vessel. So which will then lead to the formation of platelet plug. So this platelet plug, what happens is there is an activation of the coagulation cascade. So which is actually the clotting factors that promotes the formation of the fibrin clotting. So fibrinogen will convert into fibrin, which is insoluble in nature in the presence of thrombin. Thrombin. Okay. And lastly, this there will be the formation of fibrin plug or the final clot. Right. So this is basically how the blood clotting occurs. Okay. So these responses, these mechanism, that is the signal cascades, are usually the complement pathways, and there are basically three different type of pathways. First is classical pathway, which actually involves antigen antibody immune complexes, and this particular classical pathway is activated when C1q binds to the antibody, and then it will break down to C4 and C2, and then again to C3 and C5 convertases. So if the C3 and C5 convertases breaks down to C4A, C3A and C5A, it will lead to inflammation. Similarly, in lectin pathway, there is a PAM recognition by lectins. So MBL, that is mannose binding lectin, uh, when um, when binds to a carbohydrate in the presence of MASPs. MASPs is mannose activated serine proteases. So this will again break down to C4 and C2, and then again similar to the classical pathway, it will cause inflammation or lysis if C5b9 is produced. In case of alternative pathway, there is a this gets activated when there is a spontaneous hydrolysis, or there is a presence of any pathogenic substance or pathogenic organism on the surface. So again, this can also result in opsonization, lysis, and inflammation. So particularly what happens, this sequence of how the events during the sequence of responses is, firstly there will be an injury, then it will lead to acute or chronic inflammation, then it will result in granulation of the tissue, then we'll able to see the host body reaction, and lastly there will be fibrosis. Similarly, you can see from this particular graph, the sequence of responses has been, if you plot a graph between the intensity or the time. So first there will be an inflammation, as you can see in the top of the graph, on the top left side it's written. So acute inflammation will occur, then it will lead to chronic inflammation. So during the first few moments of the responses or the inflammation, there will be the production of macrophages. So macrophages will be present in high amount. Then later on, there will be vascularization. Later, FPGCs, fibroblasts, and lastly, there will be fibrosis. So, this sequence of responses you can correlate with the time as well. So, what are the factors affecting biocompatibility? So, biocompatibility is basically how compatible the biomaterial is with the host body. Right. So, while implanting a biomaterial into a body, how efficient or how what is the ability of that biomaterial to work properly in the host body is referred to as biocompatibility of the particular biomaterial. So this biocompatibility depends upon the composition of the material, like what are the properties of the material, what is the mechanical properties, what's its surface topography, including the surface texture as well as the pore size. Next is the surface ligand landscape. Then it's the degradability factor of the biomaterial. So in certain cases, like in case of a surgical suture, we usually use some degradable biomaterials so that later on when the injury heals, the biomaterial will degrade itself, right? Whereas in other cases, such as sometimes if there is certain fractures in your body, we do not use degradable um biomaterial, but we mostly try to use biomaterial made up of metals, um, so that later on it can be again recovered through surgery itself. Next is ability to resist infection. So any biomaterial, if it's implanted in the body and causes like secondary infections apart from the primary infection that is caused, primary infection or primary disorder that is present in the body, it can able to again cause any other infection, is not particularly suitable as a material for material for the biomaterial. Okay. And lastly, there is proper surgical placement. So if the biomaterial is not placed properly during the surgery, then also it can lead to some inflammation or injury. So this can again cause difficulty, right, in the proper working of that particular biomaterial. Okay, moving on, let's go through the lecture 17 recap. So in this, we first talked about what is this cell adhesion mechanism. So this cell adhesion mechanism is modulated by cell surface receptors, which are membrane embedded proteins or glycoproteins. So these cells attach to each other and as well as to the extracellular matrix through adhesion receptors. So this adhesion receptor binds specific peptide sequence of other proteins. Right. So cell attached to other cells or to the extracellular matrix through adhesion receptors. So you must remember these adhesion receptors enable these cells to bind specifically to specific peptide sequences of other proteins, right? So there is particular adhesive sites. It can be either focal adhesion, that is around 10 to 20 nanometer in range. So these include various adhesive proteins like laminin, fibronectin. Then there is close contact adhesion, that is in the range of 30 to 50 nanometer. Also, there is this extracellular matrix contact that is greater than 100 nanometer in range. So in this case, there are the adhesive sites may include collagen, enzymes, elastin, glycoprotein, then proteoglycans as well. So pre-adsorbed proteins and proteins produced by cells govern the strength and kind of cell adhesion. So the pre-adsorbed proteins as well as the proteins produced by the cell decides the strength and the kind of the cell adhesion. Like how strongly can the cell adhere to the surface or adhere to the protein. Okay, moving on. So there are particularly five classes of these cell adhesion molecules, also known as CAMs. It can be either the first classes like cadherin. Next is Ig superfamily CAMs. Ig is immunoglobulin superfamily CAMs. Then there are also mucin-like CAMs. Then there is integrins and as well as selectins. So in this particular figure, you can see all the structures of cell adhesion molecules and how they are placed in this matrix or the membrane. So if we talk about the integrin CAM, so the integrin family is composed of 24 alpha beta heterodimeric member that mediate the attachment of cell to the extracellular matrix, and there are around it is composed of 8 beta subunits that can assort with 18 alpha subunits to form the 24 distinct integrins. So you can see in this figure that there is written RGD receptor. So in case of integrins, these RGD receptors are very commonly found. So RGD is arginine, glycine, and D is your aspartic acid. So these are the amino acids receptors. So you can remember this RGD receptors are commonly found in integrins, and what is this basically RGD means. Okay, now let's talk about how you can characterize the cell adhesion. Cell adhesion can be characterized on the basis of morphological analysis or even biochemical analysis. So in case of morphological analysis, you can determine the total number of initial cells that is available for adhesion, or you can determine the surface area present for the adhesion, as well as you can determine the cell shape index, which can be calculated by 4 pi a by p square. So a is area and p is the perimeter square. So suppose if the cell shape is if the cell shape is circle, so the area will become pi r square and p will become 2 pi r, right? So if you calculate it, so pi r square by 2 pi r to the whole square, you will get around 1 by 4 pi, right? So this 4 pi is already written here. So 4 pi divided by 4 pi will be 1. So the cell shape index for a circle, for a cell which has circular shape, will be around 1. So can you tell me like what is the desired value for cell shape index at which there will be maximum cell adhesion? So usually what is the thing is for maximum cell adhesion, uh, the cell shape index should be on the lower side. That is around 1. Sorry, that is around 0. So in between 0 to 1, if your cell shape index is 0, then you will have maximum cell adhesion. Whereas if you have the cell shape index as 1, you will have it as the least cell adhesion. So basically we are determining the cell shape, and if it is around 0, then we term it as line and there will be maximum cell adhesion on the surface. Fine. Also, you can characterize the cell adhesion on basis of biochemical analysis. So you can analyze the structures of actins, integrins, then extracellular matrix, and as well as the enzymes, for example, alkaline phosphatase. So moving on to cell adhesion kinetics. So cells when present in the suspension, it is present in bulk, and at interface you can see, and then cells at surface. So there will be a end on and side on and end on adhesion. So moving on to the cell adhesion kinetics. So basically what happens is NB, that is number of cells in bulk, converts to Ni, that is the number of cells present in the interface, plus the surface will result in the number of cells present in the surface, right? So Kf, Ka and Kd. Kf is the film mass transfer coefficient, K is the rate constant for cell adhesion, and Kd is the rate constant for cell detachment. So once the number of cells that is present in the surface, in the interface gets like is is attached to the surface, it will be through this rate constant for cell adhesion. So this particular conversion will be determined by Ka, that is rate constant for cell adhesion, and if there is the detachment, that is the cell moves away from the surface, it will be determined by Kd, that is rate constant for cell detachment. So this can be analyzed by this formula, that is dNs by dt. Ka, total number of cells present in the interface, then surface is equal to Ka and B minus Ns S, and the above equation can also be written in terms of the surface coverage. Surface coverage is given as theta, which is actually Ns into Ac by As. So what is this Ac and As here? So Ac is the average area of the adhered cell and As is the total surface area. So d theta by dt, that is surface coverage with respect to time, is given as Ka Nb multiplied by Ac by As minus theta into Sf. Now what is Sf? So Sf refers to the surface fraction which is available for the cell adhesion. So this theta can be also represented in terms of percentage. So 100, 1 minus e to the power minus sf into Ka into T. So anytime if there is a question regarding determination of the surface fraction available for cell adhesion, you can use this particular equation. Okay, we shall move forward now to the recap for lecture number 18. I hope there is no question. So till now we went through lecture number 16 and 17. We basically went to the overview of it. We did not go through every detail. So let's move ahead and discuss what was being taught in lecture number 18. So in this lecture, biocompatibility of biomaterials was discussed. So what is this biocompatibility? As I already discussed here, biocompatibility is the ability of a material to perform the desired function in harmony of the host cell. So any biomaterial, when it is implanted into a body, so it is termed as biocompatible only if it has the ability to perform the desired function in the harmony of the host cell. So um, if it is not suitable or it is not uh, functioning properly in the body, so it is not a biocompatible biomaterial, right? Also it has the ability to perform with an appropriate host response in a specific application. So every biomaterial has a specific function, right? And for that specific function, there is a requirement of an appropriate host response as well. So when these two happens in harmony, it is the biomaterial is termed as biocompatible in nature. So the biocompatibility also defines the criteria for the application of biomaterials as implants or other application. So a particular biomaterial is characterized as to be used as an implant or to be used as other application is being described by biocompatibility. So biocompatibility of the materials is based on the severity and the duration of inflammatory responses. So how long the inflammatory responses occurs and what is the severity of that response. So if the response or the inflammation is very chronic in nature, so it is and it is very harsh as well, chronic as well as harsh. So this particular biomaterial is not biocompatible, biocompatible in nature, right? So you must remember that biocompatibility of materials is assessed by the severity and the duration of the inflammatory responses. So what should be the properties of these biocompatible materials? So this should not induce acute or chronic inflammatory responses. They should not prevent a proper differentiation of implant surrounding the tissues, and this biocompatible materials depends on the purpose of the implant, right? So there are several classes of these biomaterials. It may be either made up of the composition of the biomaterial may be either metals, which are usually used in case of implants and orthopedics. Orthopedics, what do you know? What do you mean by orthopedics? So orthopedics example I can give you it as osteoarthritis or knee joint pain usually. The knee joint pain, these particular kind of diseases comes under orthopedics. Then it can be either made up of polymers and used in as a body parts or in case of joints or as well as sutures. Sutures means stitching any wound or any injury in the body is actually suture. Okay. Hydrogels again, hydrogels can be seen as the material for contact lenses, tendon materials, wound healing bioadhesives as well as cartilage. Next is bioresorbable materials. So um, any resorbable material can be used as biomaterial and applied in sutures, drug delivery. So in delivery, we want these kind of materials so that when it goes into the body, uh, it can be easily absorbed or absorbed by our body and also degradable in nature as well, right? So as well as orthopedic fixation devices. So apart from this there are class of ceramics and glasses. This particular biomaterial composition is used in case of eyeglasses, tissue culture flask as well as in case of dentistry. So usually in dentistry there are ceramics like plating over the teeth. It is very commonly used, and in case of eyeglasses also. Right, so natural materials, polymers such as used in matrix as well as support. So natural material, any kind of polymer such as even silk. Silk is also used in case of sometimes for sutures also we use silk, right? Silk material. Also composites. Composites are used in fracture fixation and joint replacement. So these are the major classes of biomaterials, and as I said, how biocompatibility can be measured using the severity and the time of the inflammatory response. By studying the severity, it depends on the severity and the time of the inflammatory response. So, particularly, various techniques are available to assess these biocompatibility. First is the in vitro approach, wherein the compatibility of the materials with specific cell lines is used, and it does not involve immunological components similar to that of the body. So here we do not use any human model or animal model, rather than we use cell lines or cytotoxicity toxicity testing. So cells can be either cell lines or even various organisms grown in petri plates also, right? Apart from that there is an in vivo approach. So in this, uh, in this particular approach, we use human with a human model or even other animal model as well. So this approach gives the actual effects of the biomaterial implants within the body with respect to its immune system, right? Because we are using human to assess the biocompatibility. So in vitro analysis, we basically assess the cytotoxicity. In this, the toxic effects are assessed at cellular level. So various tests like direct contact, agar diffusion, MEM elution, as well as MTTSA are used to analyze the cytotoxicity. Other than that, leachability is one which do not use the cells, rather than we analyze the extracts. Then there is hemocompatibility. So in case of hemocompatibility, we use blood materials. We assess the interaction between the blood materials and the biomaterial, and then we see for the response. So how compatible the biomaterial is with the blood. In case of in vivo testing, there are testing models like animals, plants, and humans. So, as I said, biomaterials are implanted within the body and the effects are analyzed periodically. So, this gives the actual effects, right? So, let's discuss the in vitro methods for testing the cytotoxicity, like cytotoxicity based in vitro techniques for assessing the biocompatibility. So first is the direct contact method. So in this, what we do is we first grow cells in a petri plate or a petri dish. Then we place the specimen as in the biomaterial directly on the cells, and then we incubate it for around 24 hours at 37 degrees Celsius in an incubator. Right. So after 24 hours and incubating it at 37 degrees Celsius, centigrade, we'll then check for the cell fixation, go for the cell fixation and analyze the living as well as the dead cell. So what happens is, if the cell lives, so we can assess that the biomaterial is compatible, right? And if the cell dies, so there is the biomaterial is not biocompatible with that cell. Fine. Next is agar diffusion. So in this, we grow cells in a petri plate again, and however, in this case, the agar is poured over the cells and then solidified. Right. So again, we place the specimen directly on the cells and incubate for 24 hours and at 37 degrees Celsius in an incubator. So here also we see the for the cell fixation and analyze the live and dead cells. So the only difference is in direct contact, uh, we grow the cells over the agar surface, whereas in case of agar diffusion, we grow cells within the agar, right? So firstly we place the cells and over that we pour the our agar and let it get solidified. Fine. So next is the MEM elution. So MEM stands for minimum essential medium. So a medium called minimum essential medium is used here. So in this we prepare extract in serum free media or 0.9% saline solution, and then we grow cells in a petri plate, and later on we place the extract directly on the cells. So then we let it incubate again for 48 hours and at 37 degrees Celsius in an incubator. Again we repeat the process same as in case of direct contact and agar diffusion, that is we fix the cell and we assess the living and dead cells analysis, right? So we are left with one more in vitro technique, that is MTTSA. So what is this MTTSA? MTT is basically a dye. So the principle behind this MTTSA is that metabolically active cells produce NADPH dependent mitochondrial dehydrogenase for controlling the electron flux through the electron transport chain, right? So the assay is based on reducing the MTT. MTT is 3,4,5 dimethyl thiazole to while 2,5 diphenyl tetrazolium bromide. So in this, the MTT gets reduced by dehydrogenase. In the presence of dehydrogenase, the MTT get reduced to form water insoluble formazan. So MTT is originally yellow in color, and in presence of this succinate dehydrogenase, what happens it converts into a purple colored substance, that is formazan, and this formazan are then solubilized in solvents like DMSO, that is dimethyl sulfoxide, or even isopropanol to determine its concentration using spectrophotometric analysis. So the thing is, this particular formazan or this purple colored substance, the intensity of this purple colored substance when it is analyzed spectrophotometrically uh, is directly proportional to the number of viable cells present in the sample. So if there is if the cell is living in nature, it will obviously produce the enzyme succinate dehydrogenase, which will result in the conversion of MDT to formazan. So once this formazan is produced, you can directly assess it under spectrophotometrically. You can analyze it, and the intensity will be directly proportional to how many number of cells are alive or live in the sample. So what we do is we incubate the cells with in the presence of biomaterials in a um, in a 96 well plate, right? And then we put MTT in uh, each of the sample, and then we let it incubate for 4 hours. Later on, we add DMSO again to dissolve the formazan, and then the formazan is being read. The purple color formazan is being read spectrophotometrically at 570 nanometer. So it will be the reading will give the number of the viable cells present in the sample. So this you can compare it from the control sample that you use as the standard. So control in case of control sample, we will not incubate the cells with biomaterials. We will incubate the cells with water or any other biomaterial which has been analyzed previously and show positive result that it is biocompatible with that cells. Or you can also go for a negative control. So in case of negative control, what we do is any other biomaterial that is not compatible with the cells. So these particular positive control and negative control and your unknown sample of biomaterial will help you to assess the biocompatibility of that particular biomaterial, right? So we will move ahead. So what are the advantages of these MTT assay? So as MTT can be reduced by all the cells, the assay applies to all cell types. So do no need to worry about the cell type. This MTT can be reduced by all the cells. So this is an inexpensive as well as a reliable method, and it is simple in procedure as well as high reproducibility. However, there are certain limitations. First is resolubilization of product is required. Yes, because we have to add DMSO at the just prior to assessing it spectrophotometrically. We have to solubilize the sample with using DMSO or isopropanol, as well as there is the low metabolic activity cells required in the high cell number. So the cell number is high, so we require low metabolic activity of the cells, right? So we just finished talking about in vitro methods for assessing the biocompatibility. Now we will try to go through what are the in vivo assessment techniques. So in case of in vivo assessment, we usually assess, we go for testing the skin irritation, we go for testing the sensitization, as well as testing the implantation, again, which is a very direct method and gives the actual effects. And lastly, systemic toxicity testing. So what are the advantages of in vivo assessment? So basically, as I said, it gives the complete response or the actual response of the body, and time drive effects of biomaterials can also be studied. But the disadvantages is that it mostly requires to sacrifice the testing models and costly as well as very time taking procedure. So usually what happen if your biomaterial is not biocompatible in nature, and if you use a human body and human body as a test sample, and you implant your biomaterial in that human body, sometimes what will happen, we may also lose that person. So these tests are usually very highly supervised, and they are very costly also, as well as time taking process. So if we see what are these biological interactions in case of animal studies, so there is interaction of various regulatory, parenchymal, stromal, and facultative cell types with the implant and with each other. Next is paracrine and endocrine factors acts on the cells around the implant. There is several cell interaction with insoluble ECM, that is extracellular matrix components, as well as soluble regulatory molecules that may have been altered by the presence of the implant. Also, in the presence of implant, there is an interaction with blood-borne cells, proteins, and molecules. Right. So let's move ahead. So various biological responses include, in case of in vivo assessment, that the dead space is created by the implant, soluble agents released by the implant, example ions or polymer fragments. There is again insoluble particulate materials or debris, you can say, that are released from the implant. Various chemical interactions of the biological molecule with the implant surface occurs. Also, there is alteration in the strain distribution in tissue that is caused by the mismatch in the modulus of elasticity between the implant and the surrounding tissue, and the movement of implant relative to adjacent tissue as a result of the absence of mechanical coupling. So, if there is a relative movement, or uh, the speed at which the implant moves and the adjacent tissue moves is not compatible, contrary to each other, then there is an absence of mechanical coupling. Right. So several in vivo analysis that includes we histology and histochemistry. So in this we assess the relative number of various cell types and the amount of ECM components around the implant. Then we can go for checking the lysosomal enzymes for macrophages. Again, we can go for biochemical analysis and detect the nature and amount of alkaline phosphatases. Then we can study the immunohistochemistry that includes membrane, intracellular, and extracellular molecules. We can again go for TEM and SEM images of the cells, that is analysis of the cells at the interface and morphology through transmission electron microscopy and scanning electron microscopy. Lastly, we can study the biochemistry of the inflammatory mediators, as well as we can go for the mechanical testing, which will help us know the mechanical strength of the biomaterial. Okay. So through all these, like the whole lecture 18 emphasized on the biocompatibility. So while developing a new biomaterial, what are the factors or what are the particular properties that we need to remember? So first is the physicochemical properties of that biomaterial. Like what are the physical properties as well as the chemical composition of the biomaterial. Next is the cell biological requirements. Thirdly is the mechanical properties, then polymer chemical limitation, surgical requirements, as well as sterilization procedures. So biomaterials are expected to show better biocompatibility in both in vivo and in vitro conditions. So any biomaterial, if it show it is highly biocompatible, then only it is suitable to be implanted in the body. Okay, so we've finished the lecture 18 recap as well. Now we will go through discussion of several questions. So the first question here is, which of the following sequence of events does not occur during cell surface interaction? Your options here are A, surface water and surface protein, surface protein then surface cell interaction, C is surface cell interaction and then surface protein interaction, and lastly is none of the above. So you have to select one of the option that is not correct and which does not occur during cell surface interaction. So there are sequence of events. So first option A is surface water and then there is a surface protein interaction, or option B, surface protein and then there is a surface cell interaction, and option C is surface cell and then there is a surface protein interaction, or option number D, which is none of the above. So I will let you to absorb the question first and know what the questions ask and then try to answer it. Okay, fine. We will try to see what's the answer is. Okay. So the correct answer to this question is option number C, that is surface cells and surface protein. So why so? So what happens during the cell surface interaction? So firstly there is a surface water interaction, then there is a protein surface interaction, and lastly there is a cell surface interaction. Right. So protein surface interaction, that is the protein is already present in the surface, and cell surface interaction also includes the protein surface interaction, right? So the first option A, that is surface water and then surface protein, is a correct answer. So remember that in this question we were asking which event does not occur during the cell surface interaction. So as I just even I got confused as the first option was correct. So this particular event, that is first there is a surface water interaction and then there is a surface protein interaction, is absolutely correct as it occurs during the cell surface interaction. Next we have is the surface protein and then there is a surface cell interaction. Yes, this is also a correct one. However, your option number C, that is surface cell and then surface protein, this is not a correct option as firstly there is a surface protein interaction from option B you can see, and then there is a surface cell interaction, right? So any other question wherein you can also be asked to describe the events of the cell surface interaction in a correct way, where all these three will be also given and you have to rearrange it to give the correct sequence of events can also be asked. Okay, we can move on to the next question then. So you have your question here as you have to complete the term A and B. So A converts to B in the presence of thrombin. So you have to tell me what is this A molecule which gets converted into B molecule in the presence of thrombin. Okay. So your option here are A is fibrinogen, that is represented as A, gets converted into fibrin, which is insoluble in nature in the presence of thrombin. Or option number B, thrombinogen converts to thrombocytes in the presence of thrombin. C is fibrin, that is insoluble, converts to fibrinogen. Or lastly D, pepsinogen converts to pepsin in presence of thrombin. So pepsinogen is A and pepsin is B. So can you give me the answer? It's like even if it's a guess, you can try to think and actually give the answer. Even if it's a guess, no problem, because I'll be obviously explaining the answer and the reason behind it. Okay. Okay, so you have to complete the term A and B, or you have to say what is the term which is A and B. Is fibrinogen is getting converted to fibrin, which is insoluble in nature in presence of thrombin, or is it a thrombinogen that converts into thrombocytes, or is it fibrin that is insoluble and converts into fibrinogen in presence of thrombin, or lastly is it pepsinogen that is getting converted to pepsin? Okay, if there is no guesses, we can go further and discuss for the answer. So the correct answer to this question is option number A, that is fibrinogen is getting converted to B, that is fibrin and insoluble in nature. Right, so activation of this coagulation cascade, that is clotting factors, promote the formation of a fibrin clot. So what is this clotting factors? It is actually this is thrombin. Okay, so fibrin in the presence in the when this thrombin is activated in its activated form, then fibrinogen get convert into fibrin, which is insoluble in nature, and this fibrin which is insoluble, actually it forms fibrin plug or the final clot, right? So thrombinogen, option number B, thrombinogen getting converted to thrombin, uh, or sorry, it should be thrombocytes, is actually not the correct option. Then again, fibrin insoluble to fibrinogen is not correct. And last also pepsinogen to pepsin. This usually happens um, in the intestine, in the stomach, uh, and not in case of this hemostasis. Okay. So we will now move on to the third question. So which of the following is not involved in cell adhesion? So you have to answer me which of the following is not involved. Okay, we already know what are the adhesion sites or the adhesive sites that are been involved in cell adhesion. So you have to answer which is not involved in case of cell adhesion. So your options here are A, focal adhesion, B, intermolecular contact, C is your close adhesion, and D is extracellular matrix contact. So you have been given four options, that is focal adhesion, intermolecular contact, close adhesion, and extracellular matrix contact. So one among these four is not involved in case of cell adhesion. If you can, so this cell adhesion adhesive sites we already discussed in case of lecture number 17. If you go through your lecture number 17, you will get to know what are the cells, what are the adhesive sites that have been involved in case of cell adhesion. Fine. Okay, we will see the answer to this question as well. So the correct answer is option number B, that is intermolecular contact. What happens is various adhesive sites, I already discussed it, that is focal adhesion that ranges between 10 to 20 nanometer. This involves various adhesive proteins like laminin, fibronectin. Then there is close contacts, that is 30 to 50 nanometer uh, ranging. And lastly, extracellular matrix contacts, that is in the range of greater than 100 nanometer. So this usually involves collagen, enzymes, elastins, glycoproteins, as well as proteoglycans. So there is no intermolecular contact involved in case of cell adhesion, whereas all other three options, that is option A, option C, and option D, that is focal adhesion, close adhesion, and extracellular matrix contact. All the three are um, involved in case of adhesive sites or in case of cell adhesion. However, these intermolecular contacts are not usually does not represent the cell adhesion, right? So we can move on to the fourth question if there is no doubt. So question number 4 is, which of the following belongs to the class of cell adhesion molecules? So cell adhesion molecules are also known as CAMs. So you have to tell me that which of the following belongs to the class of cell adhesion molecules. So your option here are A, cadherins, B, selectins, C, integrins, and D, all of the above. So if you recall back your lecture number 17 again, there are we actually discussed about the class of cell adhesion molecules. So if you recall back, as I said, there are around 5 classes of cell adhesion molecules. So you have to tell me which among the following belongs to the class of cell adhesion molecules among those 5 classes of cell adhesion molecules. I'll give you 1 minute to think and try to answer the question if possible. so the correct answer to this question is option number D, all of the above. So as you can see, we already discussed what are the five classes of cell adhesion molecules. So it involves cadherins, Ig superfamily CAMs, then there are mucin-like CAMs, then there is integrins, and lastly selectins. So all the first three options, that is cadherins, selectins and integrins, belongs to the class of these cell adhesion molecules. So your correct option would be option number D, that is all of the above. Right. So you must remember that what are the five classes of these CAMs. Okay, so first it is cadherin, Ig superfamily CAMs, then there is mucin-like CAMs, integrins and selectins. Okay, let's move on to the next question. So we have the question number 5 as which of the following is or are the ways to characterize the cell adhesion. So how do you characterize cell adhesion? So your options here are A, analyzing the cell shape index, B is the number of cells available for adhesion, C is determining the rate of cell adhesion, or D, all of the above. So which among the following is the way to characterize cell adhesion? So uh, this particular information is available in lecture number 18. If you go back through your, lecture number 17. So if you go through the lecture number 17, we already discussed what are the methods or how do you characterize cell adhesion. So first option is if you analyze the cell shape index. So if you recall back, what is this cell shape index? So cell shape index is calculated as 4 pi A divided by P square. So A is your area and P is your perimeter of the cell shape. Okay. So B is determining the number of cells available for adhesion. That is what is the initial number of cells that is available for adhesion to the surface. C is determining the rate of cell adhesion, that is Ka, and lastly all of the above. So the correct answer is option number D, that is all of the above. Right. So analyzing the cell shape index can help you analyze or characterize the cell adhesion, which okay. So option A, that is analyzing the cell shape index, comes under the morphological analysis for characterization of cell adhesion. Next is determining the number of cells that is available for adhesion, or the initial number of cells that is available for adhesion, also helps in characterization of cell adhesion. Then if you determine the rate of cell adhesion, that is Ka, again it also helps in determining the or characterizing the cell adhesion. So your correct answer would be option number D, that is all of the above. Apart from this, you can also go for biochemical analysis, wherein you study the structures of actin, integrins, ECM, that is extracellular matrix, and as well as determining through enzymes like alkaline phosphatase. Okay, we will now, if there is no doubt, we can move ahead to question number 6. Question number 6 is your the in vitro analysis of the biomaterial involves assessment of. So options are A, cytotoxicity, B, leachability, C, hemocompatibility, and D, all of the above. So if you think, what is this in vitro analysis? Can you tell me what is this in vitro analysis of biomaterial? So firstly, you need to define what is this in vitro analysis as well as in vivo analysis. So in vitro analysis involves or is the approach wherein we use cells or cell lines to determine the biomaterial biocompatibility, or in case of in vivo analysis we use human, animal as well as plant models to test the biocompatibility of the biomaterial. So what happens in vitro analysis, if you recall back? So as you see, your option A is cytotoxicity. So as I said, in vitro analysis involves cell lines. So yes, we do cytotoxicity. We do assess the cytotoxicity through in vitro analysis. So if you rule out option number A, we then go through the option number B, that is leachability. So what do you mean by leachability? So leachability is actually analyzing the extracts. So we do not place the specimen directly onto the cells, rather than we place the extracts uh, over the specimen, and then we let it incubate and assess whether it is live or dead in nature. Then there is hemocompatibility, wherein we try to assess the relation between the blood and the material. Okay, so blood and the material, or the blood extract and the material compatibility testing. So all these three does not include any human model or plant or animal model directly. So this all three A, B, C and C options are involved in vitro analysis of the biomaterial assessment. So the correct answer would be option number D, that is all of the above. So you can see that in vitro analysis usually involves cytotoxicity testing, leachability assessment, as well as hemocompatibility assessment. In case of cytotoxicity testing, there is direct contact, agar diffusion, MEM elution. Actually, if you see, MEM elution is leachability testing. I think in that we use extracts, right? Okay, fine. We will now move on to the next question if there is no doubt. Okay. So your question number 7 is, the in vivo analysis of the biomaterial does not involve A. Immunohistochemical analysis, B. TEM and SEM, C. MTTSA, D. Biochemical analysis. So you have to now recall what is this in vivo analysis and what kind of assessment do we do that involves in vivo analysis. However, in this particular question, you have to tell which of the particular option does not involve in case of in vivo analysis of the biomaterial. So options here are A. Immunohistochemical analysis, B is TEM and SEM, that is transmission electron microscopy and scanning electron microscopy. Then there is MTTSA, and lastly biochemical analysis. So in case of in vivo analysis, what do we do is we use animal, human as well as plant models for testing, and then we determine the immunohistochemical analysis or even go for TEM and SEM images of the sample, as well as we go for biochemical analysis. However, we do not use MTT assay for analyzing in vivo assessment, because we this particular assay, that is MTT assay, is used in vitro analysis, right? So as you know, MTT is a dye and cells that produce succinate dehydrogenase. It converts the MTT from yellow color to a purple formazan, and this purple formazan, in the presence of, when it is being solubilized using DMSO or isopropanol, and we can then assess the sample spectrophotometrically. So the intensity of the sample or this formazan will be actually directly proportional to the number of cells or the quantity of cells that is alive in the sample, right? So MTT assay basically detects the cytotoxicity that is usually assessed in case of in vitro analysis and not in case of in vivo analysis. So the correct answer would be your option C, that is MTT assay, right? So all the other three options, that is immunohistochemical analysis, biochemical analysis, as well as STEM and SEM analysis of the cells, comes under in vivo analysis. What else do we analyze during in vivo testing of the biomaterial is, we also go and determine the histology as well as the mechanical testing also, right? So you must remember all the five, so that any other option, if it is included, you are able to give the answer. Okay, we can move to the next question. Question number 8 is, which of the following biomaterials is not suitable for a surgical suture? A is polymer, B ceramics, C bioresorbable material, and D silk. So you have to tell that which of the following biomaterials is not suitable. You do not have to tell that which is suitable for surgical suture, but you have to tell which is not suitable for a surgical suture. So, so three options among the four are correct, which is actually suitable for a surgical suture, whereas one of them is not suitable for it. So can you recall back what are the classes of different classes of biomaterial, or what are the composition of these biomaterials, and think that where can be used what kind of material? For example, in case of surgical suture, what kind of material or biomaterial is used? So I will give you 1 2 minutes to get through the question and try to answer it if possible. Hello? Yes, sir. Yes, sir. I'm good, sir. Yes, sir. Uh, Actually, I was doing from last 2 days. I was doing gram staining, um, and now I am actually going for various tests like oil displacement test. For that, I have like gone for the incubation of the organism. So once like for 4 days, I have kept it in the incubator. So tomorrow I'll be extracting the cell-free supernatants and then going for the test. Yes, sir. 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 Okay, sir. Yes, sir. No, sir. Not yet. Uh, yes, sir. Sir, actually, few of the things that is with Vishal, he said that he'll be writing those. So he asked, yes, sir, yes, sir. But I think he had a talk with Rajiv Sharma, sir. No, sir. So maybe in the next week, by Tuesday or Wednesday, Vishal said that we'll try to wind it up. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes. Uh, Sir, no, sir. He was from France, so from Bruno. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Oh, no, sir. Uh, yes, sir, because I have in this last week of April, I have to submit the report for PMIF also. Okay, sir. Okay, sir. I will talk to Pandey, sir. Okay, sir. Okay, sir. Okay, sir. Okay, sir. Okay, sir. Okay, sir. I'll prepare that report for you. Yes, sir. Yes, sir. Yes, sir. That's what I was doing through like last 2, 3 weeks in the environment. Yes, sir. Yes, sir. Yes, sir. Sir, I was also planning to propose a like abstract for RIC. So now I can at least present a review. So actually, it will again take like 2, 3 months to complete this particular work. It's not yet complete. It's the like preliminary stage. Yes, sir. Yes, sir. Yes, sir. I am working on that as well. Okay, sir. So it's in May 20 and 21. Yes, sir. Okay, sir. Okay, sir. Thank you, sir. Okay, so yeah, I'm extremely sorry as I got a call very urgently, so I had to take your 5 minutes. So now, so we were discussing about question number 8, that is, which of the following biomaterials is not suitable for a surgical suture? So our options here are A, polymers, B, ceramics, C, bioresorbable material, and D is silk. So the correct answer to this question is your option number B, that is ceramics, right? So why so? If you look into this different classes of biomaterials, so for a surgical suture, we usually use polymers, natural polymers, as well as bioresorbable materials. And silk is also used in case of surgical suture. However, ceramics is usually used in case of dentistry, um, also in case of tissue, culture flask, and eyeglasses, right? So ceramics, we don't use it in case of surgical suture. So different composition of biomaterial is used in different application, right? So we cannot use any metals in case if we are using surgical sutures, right? Because what happens in suture, we usually stitch the injury, or we stitch the wound, right? So when we stitch it, we want something uh, which have a degradable, which is degradable in nature. So here, ceramics is not usually used in case of surgical suture, whereas polymers, bioresorbable material, as well as silk are all used in surgical suture. Okay, we will now move ahead to question number 9. So again, here the question is, which of the following biomaterials is used for contact lenses? So your option here are A. Hydrogels, B. Metals, C. Glasses, and D. Ceramics. So think about what can be the properties of contact lenses. What do you want in the properties of the contact lenses? So it should be obviously uh, it is a contact lens, so it should be also help you to view the surroundings again. mm, so for in this case, it should be also non-reactive with your eye lens. And what else? So if you see option number B, that is metals, so usually we don't use metals, right? In case of contact lenses, if we use metals, what will happen? Metals are not comfortable if we to place in our eyes, right? Next is glasses. Again, glasses are also not suitable for contact lenses material. Why? If any glass broke, or if any glass component breaks, what will happen? It will eventually damage our eye, right? Again, ceramics. So ceramics we already uh, saw that in last question. Ceramics are usually used in case of dentistry. So what we are left with is option number A, that is hydrogels. So usually these lenses are gel-like material, and which has uh, like properties of soothing the eye, and it is non-reactive with the eye, as well as it does not damage the eye lens, right? So the correct answer for question number 9 would be option A, that is hydrogels. So where else do we use this hydrogel? So we use it in case of tendon materials as well as wound healing bioadhesives, and in case of cartilage, like cartilage replacements. Like if you have any injury and your cartilage is damaged, then these hydrogels are used, right? So metals are used in implants and orthopedics. Glasses, again, glasses and ceramics are used in eyeglasses, tissue culture flask, and dentistry. So the correct answer would be option A, that is hydrogels, which is a suitable class of biomaterial used for contact lenses. Okay, uh, we can, if there is no doubt, we will move on to our next question. So we have our question number 10, that is the in vitro analysis of biomaterial does not involve. So similar question we had for in vivo analysis. So your options are A, MTT assay, B, agar diffusion, C, histochemical analysis, and D, MEM elution. So again, you have to recall back what is this in vitro analysis, wherein what is this in vitro approach of assessment of biocompatibility of the biomaterial, and what is the in vivo approach, right? So if you are able to distinguish between this in vitro and in vivo analysis techniques, then you will be easily able to answer this question. Okay, so MTT assay, option A, where do we use this MTT assay? So in case of MTT assay, what do we do is, what is the procedure? In this, we take a 96 well microtiter plate, and then we place our cells over which we place this MTT dye. So if the cells are alive, that is Also, we place our specimen to be analyzed, and if the cells remain alive, what will happen is the MTT will get converted to formazan, which is purple colored solution. And so if the cells are alive, they will produce this succinate dehydrogenase enzyme, which will help this MTT to get converted into formazan. Then this formazan can be solubilized in the presence of DMSO or isopropanol and can be read spectrophotometrically to analyze the intensity of the cell, uh, the intensity of the cells that are alive. Again, in agar diffusion, so in this we pour the agar over cells and then we place the specimen on top of it. We let it incubate and then we fix the cells and analyze the dead as well as the living cells. So this particular assay, that is MTT assay as well as agar diffusion assay, you can see both of these assay determines the cytotoxicity, because at the end we are basically testing if the cells are live or dead. Similarly, in case of MEM elution also, we basically test the cytotoxicity, or you can say the leachability also we test, right? So all these three options, that is MTT assay, agar diffusion and MEM elution, are in vitro analysis techniques for the biomaterial, right? For determining the biocompatibility of the biomaterial. However, this histochemical analysis is a in vivo approach or in vivo assessment. So if you go back in case to the slide where we were discussing about the in vivo analysis, so you can see there is this histochemistry or the histochemical analysis which we perform while analyzing the biocompatibility through in vivo testing. However, in case of in vitro analysis, we have the cytotoxicity, leachability, and hemocompatibility tests, right? So we can now move forward to 11th question. So which of the following should be considered while developing a new biomaterial? Options are A. Physicochemical properties, B. Mechanical properties, C. Polymer chemical limitations, or D. All of the above. So what are the factors that you should keep in mind when you are developing a new biomaterial? So you have to assess the physicochemical properties, or you should see what are the physicochemical properties or the mechanical properties or the polymer chemical limitations, or is it all of the above? So I will give you 1 minute to think if you could possibly give the answer. So you have to select that which of the following should be considered while developing a new biomaterial. So what all things or factors you must keep in mind while developing a new biomaterial? Okay. So the correct answer to this question would be option number D, right? All of the above. So why would it be the correct answer? So when you develop a new biomaterial, you must see what is the application of the biomaterial first, right? And then you have to see what are the physicochemical properties, then the cell biological requirements as well as its mechanical properties, Then there is any surgical requirement or sterilization procedures, as well as the polymer chemical limitations. So as you see, your option A, that is physicochemical properties. So any biomaterial, you have to know what is its physical properties as well as chemical properties, right? Like what is it made up of, or what is its melting point or boiling point, or even you have to see whether it is at what temperature is it, or it functions uh, like chemically at uh, what condition it is activated, right? Then you have to see the mechanical properties. So if you are implanting a particular biomaterial in case of any fractures, so suppose, so if you are implanting a particular biomaterial, right? In case of fractures. So what do you want it? You don't want it to be degradable, right? You want it to be mechanically tough. So you have to assess the mechanical properties of that particular biomaterial as well. So
दरवाजा
according
बंद
to,
करके जाना,
one second.
दरवाजा बंद करके जाना, दरवाजा मोहम्मद.
Okay. So next is polymer chemical reaction, right? So polymer is any kind of natural polymer or other polymer. Basically, suppose if we use in case of suture, right? So this polymer chemical limitation also we have to assess at not just the properties but also the limitation which the biomaterial possess. So while developing a new material, a new biomaterial, you must include all these kind of, or all these factors should be considered. So apart from physicochemical, mechanical and polymer chemical limitations, you must also see for cell biological requirements. Then you can also see for the surgical requirements, whether the biomaterial that you are using is actually to be used or applied in case of any surgery. Or lastly, sterilization procedures, right? So if this biomaterial that you are using requires any sterilization, or if um, like does it have the ability or does it infects when it once it is inside the host body, right? So I think this question is clear, so that we can move forward to the next one. Okay, let's discuss the question number 12. So what is this question? The classical pathway of the complement system is activated. Options are: A, when C1q binds to antibody attached to the antigen; B, when mannose binding lectin binds to the microbial surface oligosaccharides; option C is when spontaneous hydrolysis of the internal C3 thioester bond, and also there is a contact with various structures on the microorganisms and other foreign surfaces; or is it D, none of the above? So if you go back and remember the complement system pathway, so it has basically three pathways. One is the classical pathway, another is the lectin pathway, and lastly is the alternative pathway, right? So you have to tell me, you have to answer the question as, in case of classical pathway of the complement system, how does it get activated? Okay, so yes. So in case of classical pathway of the complement system, how does it get activated? Is it like when C1q binds to the antibody attached to antigen? Or is it when MBL binds to the microbial surface oligosaccharides? Or does it get activated when there is a spontaneous hydrolysis of the internal C3 thioester bond, as well as there is a contact with various structures on the microorganisms or other foreign surfaces? Or is it none of the above? Okay. So we will try to answer this question. So the correct answer would be option number A, that is the classical pathway of the complement system gets activated when the C1q binds to the antibody attached to antigen. Right. So if you see this particular photograph or picture, so in here you will find all the three pathways, that is the classical pathway, lectin pathway and alternative pathway. So in case of classical pathway, what happens is it gets activated when there is a C1q binds with the antibody, right? Whereas in case of lectin pathway, when MBL or mannose binding lectin, MBL, right? Mannose binding lectin binds with the carbohydrate moiety. As you can see in the option number B, it is given when mannose binding lectin binds to microbial surface oligosaccharides. So oligosaccharides are nothing but it is a type of carbohydrate only. And in case of alternative pathway, you can see there is a spontaneous hydrolysis of or there is a contact with the pathogenic surfaces. So your option C, that is when spontaneous hydrolysis of the internal C3 thioester bond and contact with various structures on microorganisms and other foreign surfaces. So option A here is your classical pathway and B is the B is the process by which your lectin pathway gets activated, whereas C, option C is your third, that is alternative pathway activation process, right? So classical pathway, lectin pathway and alternative pathway. So three pathways of the complement system. However, in this particular question, you were asked to determine what is the particular pathway or particular process by which your classical pathway gets activated. So you can see from the figure itself that the C1q, when it's bind to the antibody, then your classical pathway gets activated. Fine. I hope this question is clear so that we can move on to the next question. Okay. So question, is there any doubt? So you can ask about the previous question. If not, question number 13 is which among the following involves the factors that affect host response to biomaterials? So option A is composition of the material, B is degradability, C is proper surgical placement, and D is all of the above. So your question here is which among the following involves the factors that affect the host response to biomaterials? So obviously you will also have to think about what is this biocompatibility and how does biomaterial what happens that leads the biomaterial to evoke a host immune response, right? So if you think about biocompatibility, so what is biocompatibility? So biocompatibility is the ability of the biomaterial to be in harmony or to function in harmony with the host body, right? And to be fully able to function as well as does not cause any other or any chronic or acute inflammatory response. So if you see your option A, that is composition of material. So does it affect the host response of to biomaterials? So yes, if suppose if the biomaterial is made up of certain composition or certain class of biomaterial that it evokes a immune response or it is not compatible with the body, then of course it will affect the host response, right? Second is degradability. So if you want the particular biomaterial to be degradable or non-degradable in nature, and does how does it suits the body, so this particular factor also affect the host response to the biomaterial. Your option C is proper surgical placement. So if you place your biomaterial improperly or implant the biomaterial improperly, there are chances that it will evoke a immune response, and it will damage the tissue, and it will evoke the immune response. So what will be the correct answer to this question? Okay. So the correct answer would be option D, that is all of the above. Right. So what are the factors affecting biocompatibility apart from all these three? You again have your mechanical properties, then there is surface topology, or that is the surface texture or the pore size. Then you have surface ligand landscapes. Again, the ability to resist infection is one of the factors that affect the biocompatibility. So if your biomaterial um, causes certain infection when once it is inside the body, or even once it is over the surface of your body, then it is not biocompatible, right? So you must remember all these points, that what are the factors that affect the biocompatibility. Fine, I think we can move next if there is no doubt. Okay, so the next question is, the DLVO theory demonstrates the relationship between option A is cell distance from the surface and cell size. Option B is particle distance from the surface and repulsive and attractive interaction energies. Option C is cell size and electrostatic interaction energy. And D, cell size and van der Waals interaction energy. So I did not discuss this DLVO theory, so I tried to put it in a question here so that we can cover most of the concepts, whatever was being taught in week 6. So what is this DLVO theory basically? So you have been asked here that what is this DLVO theory demonstrates the relationship between cell distance or the particle distance from the surface and cell size, or the particle distance or the cell distance from the surface and repulsive and attractive interaction energies. C, cell size and electrostatic interaction energy. And D is cell size and van der Waals interaction energy. So if you see, the correct answer would be, so okay, let us discuss what is this DLVO theory. So DLVO basically are the person's name, that is Dejaguin, Landiu, Vervi, and Overbeek, right? So this is basically DLVO, and this theory demonstrates the relationship between the particle or the cell distance from the surface and their repulsive and attractive interaction energy. So repulsive energy involving electrostatic interaction, and attractive one's example van der Waals interaction energies. So both repulsive and attractive interaction energies relationship with the particle size, particle distance from the surface is given by the DLVO theory. So this is basically based on the assumption that the electrostatic double layer forces and the van der Waals forces are independent and therefore can be superimposed or added at each interacting distance for two particles, right? So it is often a good estimate for intersurface forces for surface separations down to about 5 nanometer. So you have to remember just that what is this DLVO theory basically demonstrates the relationship between. So the correct answer is option B, that is particle distance from the surface, and repulsive, that is electrostatic, and attractive, example van der Waals interaction energies. So in option A, you can see it's given cell distance from surface and cell size. So we do not discuss about cell size in case of this TLBO theory. Also C and D, similarly, there is this term cell size, which usually is not given by DLBO theory, right? So uh, can, if there is no doubt, we can move to the last question of today's session. Okay, so we have question number 15, which is which of the following is true for biocompatible materials? So your options are A, do not induce acute inflammatory response. B, allow a proper differentiation of implant surrounding tissues. C, depends on the purpose of the implant. And D, all of the above. So you have to tell which of the following is true for biocompatible materials. So you have to basically see what are the properties of what should be the properties of a biocompatible material, right? So which of the following is true for biocompatible material? So A is that biocompatible material do not induce any that it should not or do not induce acute inflammatory response. B, allow a proper differentiation of implant surrounding tissues. C is that biocompatible materials depends on the purpose of the implant. And lastly, you have option D, that is all of the above. So, can you guess what should be the correct answer? Okay. So, for any biocompatible material, what do we do is it should possess certain properties. For example, your option A, that is, it do not induce acute inflammatory response. So yes, these any material which is highly biocompatible, that is, it is it has the ability to perform in harmony with the host, right? So these biomaterials should, when they do not induce any inflammatory response, we can term it as a highly biocompatible material. So your option A is correct. So option A is true for biocompatible material. If you go for option B, so it allows a proper differentiation of the implant surrounding tissues. So it should not block the tissues or it should not cause any damage to the implant surrounding tissues. Rather than it should allow the proper functioning or the proper differentiation of even the implant surrounding tissues. So any biocompatible material should also allow a proper functioning or proper differentiation of the surrounding tissues of the implant, right? Option C is depends on the purpose of the implant. So yes, any biocompatible material, if we are to it to be used on the surface of your body, so sometimes in case of sutures, we want it to be degradable in nature. If sometimes if you want to use it in case of fractures, we want it to be high in mechanical strength. So any biomaterial uh, should also depend on the or its compositioning or its physicochemical properties should depend on the purpose of the implant, right? So all these three A, B, C options are actually correct or is true for biocompatible material. So the correct answer would be your option D, that is all of the above. So a biocompatible material should not induce any acute inflammatory response, as well as it should not induce any chronic inflammatory response. Right, not just acute but also chronic inflammatory response. Next is it should allow a proper differentiation of implant surrounding tissue. So it should not prevent uh, a proper differentiation of the implant surrounding tissue. It should not hinder the differentiation. Option C is it should depend on the purpose of the implant. So in case of if we use in case of for fractures, if I am going to use any degradable material, then it is not suitable, right? So it is not biocompatible to the host. For example, if I am trying to use any suppose biodegradable material in case of dentistry, if I want to have a tooth cap, right? So in that case, I don't want the biomaterial to be degradable, to be easily degradable in nature. So obviously, the biomaterial should be depending on the purpose of the implant. So the correct answer for this question is option D, that is all of the above. So this concludes the end of your session 6, your problem solving session 6. I hope all the questions were easily understandable. And if not, you can again go back and look into these slides, and as well as you can access the YouTube video also, right? So let me show you. So you can access these YouTube videos from the problem solving session. There is a particular tab in case in Swayam portal. Through that you can easily access these videos as well as the slides that has been discussed in today's session. So that particular, I can share, let me just share. So course, this is your course portal, right? So in the left hand side, you get this problem solving session option. So in the left hand side, this problem solving session option is there, and when you click on that problem solving session, you will be redirected to a link from where you can access all the videos as well as the slides, right? I'll show you the, so this is that particular Excel sheet. I hope it is visible at your end as well. So from this Excel sheet, when you click on that problem solving session tab, it will redirect you to the Excel sheet from where you can access all the YouTube links as well as the presentation folder link for all the weeks, right? So we completed week 6, and then we are left with 2 weeks, that is week 7 and week 8 course contents. So we can meet on to the next session and discuss about the week 7 course content, right? So thank you all for joining the session. We may meet in the next Tuesday live session, that is from 7 p.m. to 9 p.m. You all can leave now.